I will start in a few minutes. I will start in a couple of minutes. Right, so I'll share my slides. Uh, okay, so this is the week 10 of the live session of this course Structural Biology. Here I'll be covering the videos of week 10, which mainly involve molecular dynamic simulations. So in the previous class, we had seen the use of different softwares in visualizing a protein in 3 dimensions. Now we will go into molecular dynamic simulations. So molecular dynamics, or MD, is a process to study and analyze the dynamic nature of a system. The tool to perform molecular dynamics is computer simulation, which is the process of mathematical modeling performed on a computer, which is designed to predict the behavior or outcome of a real or physical system. So suppose you have a real system like a protein, you can perform different experiments on it, like NMR, cryo-electron microscopy, X-ray diffraction, and get experimental results, which will help you get an idea about the three-dimensional structure of the protein. So this is the way we have seen so far. The other way that can be done is that a model can be developed which mimics the real system, and this will be called the model system. And on this model system, we can perform different simulations. Like suppose the a protein is not a static molecule. It is dynamic. It can move about. It can fold in different ways. So all this dynamics cannot very easily be studied by experiments because they are on a very small time scale. But if we can create a model system of the protein, we can carry out the dynamic simulations in a time scale which we can observe, and these will give some results. And we can also use the model system to develop some hypothesis. So suppose we have a model system. We saw in the previous class how we can visualize the protein in 3 dimensions. So we can create a model system and visualize it in 3 dimensions and develop some hypothesis. Like we may say that this protein will bind with a drug, say in this region which is highlighted. The drug will, so the protein is this green and blue region, and this red is some drug that is binding with it. So we can hypothesize that the drug will bind here, and so based on that hypothesis, we can make some predictions. And then we can compare the results of the simulations with the experimental results and also the prediction that we have done. And so comparison of all three will help us improve the model. So suppose the prediction that we have is not matching with the experimental results. That means we have to improve the prediction. Or suppose the simulation result is not matching with the experimental result. That means our model is not good and we have to improve the model. So basically, what we are doing via experiments, similar things we can do computationally, and that is called molecular dynamic simulations. So what it can do is that firstly, it can calculate the time-dependent behavior of a molecular system. So what I said, the time scale of movement of a protein is too small to be studied experimentally. But using the simulations, we can see the time-dependent behavior of the protein. Then it can provide detailed information on the fluctuations and conformational changes of the proteins and nucleic acids. So each side chain of each amino acid can fluctuate with time and adopt different conformations. And suppose in the multiple conformations, only one of them is the active conformation, which is binding with the drug or the enzyme. So all that information can be gotten from molecular dynamic simulations. And it can also be used to investigate the structure, dynamics, and thermodynamics of biological molecules and their complexes. So structure we could also have gotten experimentally, but we can additionally get it theoretically and compare them. So suppose you have a molecule which is not forming a very good crystal. So the experimental results are not good. So we can improve, get a better idea of the 3-dimensional structure by using this model system and observing it computationally. And the dynamics, I already said, because of the time scale of the dynamics, it is much easier to observe them through molecular dynamic simulations. And the same goes for the thermodynamics. So here an example is given of protein folding. You have a protein here, which initially has an orientation like this, where there are, so the native state looks something like this. But initially, when it starts folding, all the native helices have not formed. So if you see the time scale at 0 microsecond, it has a state like this, where there is one long helix, but the other region, there are short helices, there are a lot of loops. Now, as you go to 18 microsecond, another helix has formed. So the native helices are represented by blue. So by 18 microseconds, 2 of the 5 native helices have formed, which are shown in blue here. Then as you go to 18.9 microsecond, another 2 helices have formed. So now you have 4 of the 5 native helices, and now slowly the loops begin forming. You can see there is a loop here in the native structure. There is a loop here. So slowly as the time goes, from 18.9 to 19.6, the different loops form. And finally, by around 20 to 25 microsecond, you have the final native structure formed. So you can observe this entire process by molecular dynamics simulations. Yeah, so as I was saying, you can observe this entire process through molecular dynamics simulations. So at each time scale, what is the structure that the protein has adopted? This can be studied using this molecular dynamics simulations. And you can see these are very small time scales, like between step 2 and 3, it's just 0.9 microsecond. This small of a time scale you cannot observe in either NMR or cryo-electron microscopy or X-ray crystallography, any of the experimental techniques. You can only observe this computationally by MD simulations. So here, like some of the features of MD simulation is shown. So the protein molecule is here. It is surrounded by something which is called a simulation box. So simulation box is basically around the protein molecule, you add solvent molecules, because a protein is not going to be isolated. It's going to be in some solution. So you will add different solvent molecules. You will add a region where the force fields will work. I will discuss the force fields later. And you will add other nearby protein molecules. So this region is called the simulation box, within which region all the calculations are going to be carried out. And there is one benzene molecule shown in red here, which is binding to the protein. So before any molecular dynamic simulation, this simulation box has to be created. And in the box, we have to specify how many protein molecules are there, how the solvent molecules are solvating the protein. So there are different solvation models that also I'll discuss. And what are the forces, interatomic, intraatomic forces acting within the protein, with the protein and solvent, between two protein molecules. All that will be specified within this simulation box. Now, after the simulation, you can get different data. So the benzene molecule shown in red here, which is binding to this region of the protein, can have different orientations, and the RMSD of the orientations can be obtained. So basically, molecular dynamics simulations will give you a snapshot of the binding event at different times. So if you give intervals of 10 microseconds, so at every 10 microsecond, it will give you a snapshot of the interaction. So if you just see the RMSD of benzene as it is binding to the this region of the protein, so wherever it is binding, the protein will also reorganize to accommodate the benzene. So there will be a RMSD in the amino acids in the binding region also. And also from these simulated structure, the delta G of binding can be calculated. So you can see that here it is an energy surface is shown. So depending on the color of the energy surface, mostly you can, this region, the main binding region, the energy is around minus 17.3 kilojoule per mole. So this is the binding pocket, because the interaction of benzene with the protein is highest. So the thermodynamics can also be obtained by this computational program, because you are specifying all the force fields. You are specifying the interatomic, intraatomic interactions. So whenever additional interactions are forming, like between benzene and protein, they can be calculated and they can be mapped. So you can see here there is a blue envelope, and blue is relatively weak binding energy of minus 5.7. So rather than that, the strongest binding is happening here, where there is a red envelope. That is the binding pocket. Molecular dynamics simulation can also give you an idea of different binding pathways. So here you have the protein. This is the binding pocket. So there can be different pathways by which the benzene is approaching the binding pocket. That can also be captured, because the molecular dynamics simulation is capturing snapshots of the structure at different times. So by putting all the snapshots together, it can be predicted the different pathways of binding, which is, so you see corresponding snapshots in the vicinity of the binding pocket is revealing that there are 4 binding pathways shown here as path 1, 2, 3, and 4. And here you have a comparison. So basically the blue bar is the delta G binding predicted by the molecular dynamics simulation, and the red is the experimental. So you can see the experimental and the simulated values are very similar for all the ligands, benzene, indole, they are all very similar, whether you're calculating the thermodynamics by the molecular dynamics simulation or by experiments. And we will go into the folding pathway later in one of the later slides. So why is this molecular dynamics simulation important? In some cases, experiment is impossible. Suppose we want to study what is happening inside the stars, or we want to predict the weather. We can't do experiments inside the stars. Or if the experiment is too dangerous, like simulating a flight or an explosion, these are 2 dangerous experiments and they will much better be visualized in a computer simulation. Or the experiment can be very expensive, like simulating high pressure or wind channel. So it is not cost effective to do these experiments. Or sometimes, like I said, the time scale is too small. So it is said the experiment is said to be blind because the property cannot be observed in that small time scale. So if the time or space scale is very small and the experiment cannot observe on such small scales, then the computer simulation comes into use. So experiments, as you know, we have seen NMR, we have seen scattering angle from X-ray. This is basically showing cryo-electron microscopy. This gives us a set of data. And from simulations also, we get a set of data. They are correlated. So if the experiments are giving good, we can get data from both the sources and we can compare them. If the experiment is giving good data, we can use that data to improve our model system, and that will in turn improve the simulated results. Or suppose the experiment is giving poor data, then we can use the simulations to get a better idea of the structure, which the experiment was not able to give us. So they kind of support each other. And as you can see, you can have in the 3-dimensional structure also, there are different levels of resolution. You can have a coarse grained structure of a protein, which looks something like this. You can have a fine structure where you can see each of the secondary structures very clearly. And you can have quantum mechanics level structure where there is atomic level resolution. So as you go from right to left, spatial resolution decreases. That means you are going into angstrom level territory, which cannot be seen by experiments. And similarly, time scale is also, when you go from left to right, time scale is also decreasing. So the time scale accessible by experiment is actually these time scales, like suppose rotation of one of the side chains, it cannot be observed by experiment. So the accessible time scales are actually more towards this side, where you can see the motion of the entire. So here there are 4 domains: orange, green, blue, and purple. There are 4 domains forming this quaternary structure of the protein. The movement of the domains can be observed by experiment. So accessible time scale, as we go from left to right, the time scale in experiment is accessible. At the left, the time scale is totally inaccessible by experiment. It is too small of a time scale. But as you go towards the right, the movement of the domains can indeed be observed by experiment. So again, this is just showing you the different experiments with increasing resolution. So the least resolution is from electron microscopy, EPR, and the resolution as you go towards cryo-electron microscopy and NMR, the resolution of the experiments increases. So basically, that is it, that some timescales are not accessible by experiments, and that is where the simulations come into effect. So now there are two kinds of experiments basically. One is in vivo, which means in life. So the experiment is taking place inside the living organism. We have seen it in short before, how the gene of interest can be cloned inside the plasmid, and it can be used to get multiple copies of the recombinant DNA. Then we can purify out the DNA that we want by electrophoresis, and we can study the structure. And same for the, or we can use the DNA to generate the protein that we want, and then separate it out by the gel electrophoresis. So that is in vivo, that is in vitro, because it is happening in a test tube. In vivo is where it is happening in a organism. So you are not extracting it out from the organism, you are studying what is happening inside the organism itself. But in this class, we will be focusing on in silico, which is experiments performed in a virtual setting, that is a computer by virtual simulation. So in silico is an expression used to mean performed on a computer or via computer simulation. And in silico studies have been used widely to predict how the drugs will interact with the body and with pathogens. For example, a 2009 study used software simulations to predict how certain drugs already on the market could treat multiple drug-resistant and extensively drug-resistant strains of tuberculosis. So this prediction was done completely by a computer. So computers can actually predict that which drugs will be very effective. Of course, you have to do experiments to prove the predictions, but the initial prediction will help you narrow down the list of drugs you are looking at. So now in molecular dynamic simulation, so in an experiment, we first prepare the sample. We put the sample in some measuring instrument, which can be the NMR spectrophotometer or the X-ray diffractometer, and we measure some property of interest for a certain time interval. In NMR, it measures the chemical shift of the respective nuclei. In X-ray diffraction, it measures the electron density and so on. And finally, we average to minimize the noise and get a clear spectrum. In molecular dynamics, the flowchart is like this. First, we prepare our system of particles, which mimics the actual sample, which can be a protein. Then we solve the equations of motion. So basically, these particles, in our case, let's consider it a protein, they are dynamic. So with each of the protein molecules, there it will have a certain position and velocity, and it will follow the Newton's laws of motion. So we have to solve those to extract the position and velocity of each particle at a certain time. And then we have to use statistical mechanics to extract different properties of the system. So the molecular dynamic simulation, the flowchart will follow like this. And there are different scales for molecular dynamic simulations. If we want to look at bond stretching, it is in the femtosecond time scale. If we just want to look at the, so for stretching of individual bonds, the time scale is between picosecond to femtosecond. If we want to look at the folding of alpha helix, it is between microsecond to nanosecond. And if we want to look at the folding of the entire protein from completely random coil to the native structure, it is a much larger time scale of second to millisecond. So to look at different properties, there are different time scales. So now we will look at how a folding of a protein is studied. Basically, instead of studying the protein, we study unfolding of protein, because we have the folded molecule. While carrying out molecular dynamic simulation.
Ma'am,
Yes.
ma'am, can you move back to the previous slide? Ma'am, can you explain this thing once more?
this flowchart?
No, ma'am, not flowchart. This molecular dynamics time scale.
So different movements have different time scales. So if you want to study the stretching of individual bonds, you have to study between picosecond to femtosecond time scale. If you want to look at the vibration of proteins, so proteins as a whole, it can move. So the movement of the whole protein, that is the elastic vibration of the protein, will fall between a little less than nanosecond to a little less than microsecond. So these are the time scales of different motions of the protein. And when we are studying something computationally, we have to give the time scale in which we want to study it. So if we want to study bond stretching, we have to give this time scale. So accordingly, we have to specify the time scale in the software that we are using so that we can look at that particular motion.
So means how we will know that which time scale we will require means.
So these have already been derived actually. I guess what people did is that they kept it at different time scales. So maybe they initially kept much larger time scales and saw that they could not see the bond stretching. So they must have tried different regions of different time intervals and to find out the region of different motions.
So for any or chemical compound, the time is same only.
Um,
Or it
Actually,
varies.
this class we will we are mostly focusing on proteins because they are the most dynamic molecules. So for proteins, these are the values. I don't know about the values for other biomolecules. Normally, you would see smaller biomolecules time like smaller molecules like don't consider biomolecule. Like suppose there's an individual amino acid, then the time scale will be much less than a protein. Larger the molecule, larger the time scale that it will take to move. So you can't say that time scale is same for all molecules, but this is the general time scale for a protein of say more between 200 to 1000 amino acids. So a general mid-sized protein, this is the general time scale.
Ma'am, means so for any protein to do the simulation, suppose if I want to do the simulation, then the general means I will put the simulation according to this time scale only.
Yes.
Or I have to read many articles and then I have to see that what the protein which I have chosen. Means by reading the articles, I have to look with the protein I have chosen has certain time intervals or not, or this in general.
No, this is the general for most proteins. Some proteins may be exceptions, then you have to read articles, but this is the general for most proteins.
And these solve equations of motion means the scientists have solved the equations of that particles and then they derive this flowchart.
Actually, the computer does this for you because there are so many atoms, so many molecules, you cannot physically do it by yourself. There are computer softwares and programs which do this for you.
For RMSD values.
Yes.
Okay.
Yeah. Okay. So basically, in molecular dynamics simulation, we get an idea about the folding of the protein. So first we have to create a model of the protein structure computationally. So a molecule is described as a series of charged points or atoms linked by springs, that is the bond. So we have to specify, say we are starting with the folded structure of the protein. Then we have to specify the position coordinates of each of the atoms and their relation with other atoms. So the bond distance and everything, all that has to be specified. To describe the time evolution of bond length, bond angle, and torsions, and non-bonding van der Waals and electrostatic hydrogen bonding interactions, etc., a force field is used. So as time goes and the protein moves, the position of the atoms will change and also the bonding and non-bonding interactions can change. So normally it is the non-bond. So suppose while the protein is moving, two atoms are coming closer together, then that bonding in energy will be greater. Or suppose they are going too far away, then the bonding energy will be less. So as the protein moves, position also changes and these bonding energies also changes. And in order to calculate that, a force field is used, which is a collection of equations and associated constants designed to reproduce molecular geometry and selected properties. So basically, the force field, we'll come into that in a few slides. The force field equations help you calculate the change of position and bond properties as the molecule moves. So the first thing is to build a molecule, a model system from the actual system by giving the positional coordinates and the distance between bonds. And then to study the evolution of the coordinates and the bond length and everything with time, a force field has to be specified. So now we will.
In that case, we have to means we have to put some force field, means we have to put some values regarding the force field.
Yes.
Ma'am, so how we will know that in which force field the protein is working nicely?
So that also, there are certain rules for that. So suppose your protein, you want to study the structure of the protein in a polar solvent. So you have to take a force field which will mimic the polar solvent. So the goal is to mimic the experimental conditions. So accordingly, different force fields have to be chosen. So it depends completely on that. Because if you want to study the structure of the protein in a crystal, you have to study a force field which is restraining the positional coordinates, because in a crystal the atoms will not move much. So you have to define a force field which will not allow the atoms to change their positions very much. So depending on the condition in which you want to study the molecule, you have to define a particular force field.
Mostly we use polar solvent only, means we use H2O because we are trying to mimic the conditions of our cell. But is it true that we can use non-polar solvents and others?
Yeah. There are options. Like if you look at the simulation softwares, molecular dynamic simulation, there are options of many solvents. You can use acetonitrile, you can use chloroform, any solvent you can use. That is the beauty of this, because in experiment, suppose the protein is not soluble in chloroform, but in simulation you can do by giving a force field which mimics chloroform, and it will give you the structure of the protein in a chloroform environment, even though experimentally you cannot get that because suppose your protein is not soluble in chloroform. So that is another way where simulation helps when experiment fails.
Ma'am, so chloroform, the bonding energy will give means according to chloroform, and the surroundings we will just control, means we will give the chloroform also, means the bond length and everything will be same as the chloroform, and in place of H2O or in place of polar solvent we are giving chloroform as the solvent.
We are giving chloroform as the solvent and the environment around the protein. So when chloroform is solvating a protein, it will create a kind of solvation shell, right? So we will mimic the force field in such a way that we will give such a force field that will mimic that solvation environment of chloroform.
Yes, ma'am. Sorry.
Yeah, sure.
So, ma'am. So means we can give
like
like that way only the force field.
Yes, yes.
So, ma'am, for that regarding a protein, we have to study the means actually my thesis paper is on this only. So means
Okay.
for doing the simulation, we need to study papers so that um, I can see the conditions in which my protein is working.
Yes.
Or without studying also we can put the simulation of means of the protein.
See, you have to have a basic idea because there are many force fields. So, which system you want to study, you won't find paper on that exact system, but if you go through literature, you will find papers of similar systems. And that paper, it will be that they have tried 4 or 5 force fields and they will say which force field has given the best result. So like that you have to do if you want to choose the best force field. If you're not doing any experiment, suppose you just want to choose the best force field which will give a optimum structure for the protein, then you have to look at literature to choose the force field.
Okay, ma'am. And I mean, in the case of these 3D structures, we will perform this from different databases like AlphaFold and Swiss model. Or
Yes, you can put the amino acid sequence and get
yes,
the
ma'am.
3D
Yes, ma'am.
structure from AlphaFold. Or you can, suppose your protein structure is similar to some deposited PDB in the protein data bank, then you can download that PDB structure and modify or mutate it to get your protein structure. So that can also be done. So yeah,
Ma'am, pardon what you have said.
so you know how you can download PDB structures from protein data bank,
Yes,
right?
yes. Mm-hmm.
So you can open that structure in a software like Discovery Studio or WinCoot, and you can mutate residues. And so suppose your protein is a mutated protein of a currently available structure which is already there in the PDB. Then you can download that structure from the PDB and mutate it according to whatever is your protein, and you can get the structure that way also.
But then we have to perform this site-directed mutagenesis and all. Means
I'm saying that if your protein is a mutated protein.
okay. Ma'am, how to
So
know that a protein is mutated or not? Is there any means? Is there any terminology or written in the or write up in the structure means in the PDB that my protein is mutated one?
yeah, like in any structure in the PDB, with that, there is an associated publication. So if you look at the publication, you can find out that whether the structure deposited is the original structure derived from the organism, or they have somehow mutated it and then deposited that structure. So
In
if
the
you just go to that publication, it will be mentioned.
and sometimes in the PDB in the bracket also when we search like uh, something like Merinobacter lipolyticus and bracket in the bracket also it's I think written that it is a
Ha 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 ha.
mutated strain.
Ha. My main
Okay.
point is it all the information is available in the PDB. So yeah, that won't be a problem.
Okay.
Okay. So as I was saying, you have to specify the different bonding interactions. So there can be bonded interactions which include stretching of bonds or bending of bonds. There are two equations associated with that. With the stretching, you have this equation. Energy associated with stretching interactions is half of the stretching constant. And then you have Rij minus R0. So R0 is the equilibrium bond position, and Rij is a position that when the molecule is moving about, the bond can lengthen or it can shorten. So these changed lengths are the Rij. So the deviation from the equilibrium position whole square. That is the energy of the stretching of the bond. And energy of bending of the bond is also similar. Here, instead of the distance, the angle is used. So you have 3 atoms and there is an equilibrium angle between them, which is theta 0. Now, while the molecule is moving, this equilibrium angle can increase or decrease. That is your theta ijk. So the deviation from the equilibrium whole square, that will give you the energy of bending.
From how to calculate this? How to calculate
Sorry?
this? Hello. Hello.
Yes.
Yeah,
I can hear you.
How to calculate this Kij? This will be in this vector form.
No, Kij is just a constant. It is like
Not
so if
Kij.
you have
Sorry, not Kij. It's Rij, means the length. You have said that
this,
compressed and stretched one.
Yes.
So how will you calculate that?
So this is again done by the software itself. There are hundreds of bonds, right? You cannot manually, and those hundreds of bonds with every microsecond, they are expanding, they are contracting. All that can't be done manually. So this is included in the computer software itself. If you suppose you are doing a simulation of 20 nanoseconds. So in that 20 nanoseconds, how many Rij values there can be, the computer will only simulate it and calculate the total energy like that.
Okay. Means of the stretched protein, total energy of the
stretching, and similarly for bending. Yeah. None of these, so all these simulation methods are done by the program. It cannot be done manually because there are so many bonds and so many angles.
So what is the need of performing this stretched energy and bended energy?
So when you are when molecular dynamics simulation is happening, you are going from folded to unfolded. The bonds are stretching and the angles are changing. So you have to calculate the total energy. So so there are 3-4 more energy terms. You add up all the energy terms to get the total energy. What the computer will do is it will minimize the energy, and your native structure is the minimum energy structure. So each of the energy terms are calculated like this to get the total energy term, and the computer will go through all the possible conformations to find the minimum energy conformation, which will be your folded protein conformation.
So after having this energy, they will by themselves only, means the computer by itself only will provide the energy, and then we have to put that energy in the simulation, and then the unfolding of the protein will start. Is it like that or something?
No. See, basically, so we start with the folded protein. And we give a certain command to the computer. We say that the temperature, so for unfolding, we have to give a high temperature. So we simulate a high temperature environment. Then all the atoms will move, the bonds will change, angles will change, and you will get many different states of the protein. And each of the states of the protein will have a different energy. The computer will then search between the different energy states to find the state which has the lowest energy, and it will say that this is the folded structure. So sorry, you start with the folded structure of the protein, you give a high temperature, and it will be completely unfolded. Then you remove the high temperature and allow the unfolded state to adopt different conformations. So the unfolded state can adopt, say, 100 different states. All of them have different energy. So the computer will go through all the states and find the state which has the lowest energy, and it will say this is the folded state. And while this is happening, while from the completely unfolded state it is going to the folded state, it will take different snapshots. So this slide was there. This is from unfolded, it is going to folded. At different time intervals, the program will take a snapshot of the structure of the partially folded protein. So looking at these snapshots, you can understand the process folding pathway. So calculation of the energy is important, because only then it will be understood that the lowest energy state is the native state, and all the state. So as you go from left to right, the energy will slowly decrease as you go from the unfolded to the folded state. That is why the calculation of the energy is important to find out this folding pathway, that by what pathway it is folding.
Ma'am, but that folded, we are providing the computer the folded protein.
Huh.
So how it will go from unfolded to folded? Means it will go from folded to unfolded only, but it will give the, yes, ma'am.
So the initial command we give is for high temperature. So the native will completely unfold and become the unfolded protein. After that, we start the molecular dynamics simulation, where we slowly allow the unfolded protein to fold back into the native state. And this is what we study.
And the folded from the folded in which we do folded to unfolded, like unfolded to folded we do in simulation, but folded to unfolded where we do that thing.
Folded to unfolded, we don't study that. We just give, there is a command to give for high temperature. As soon as we give that command, the folded state becomes, see, unfolded state can be of many different types, right? You cannot have, there is no unique unfolded state. So folded to unfolded can be multiple pathways. So there is no point in studying that. What is more important to study is the unfolded to folded pathway, because then we will get all these intermediate regions. So you can see, if we are coming from unfolded one to the folded, there are several intermediate regions. And same for the other unfoldeds also. So it is always better to study unfolded to folded. But the problem is we cannot start with unfolded because we don't know which unfolded to start from. So it is always easier to from the NMR and cryo-electron microscopy, we will get an idea of the folded structure, and we can give the command for high temperature, which will unfold the molecule. And then from the unfolded molecule, we can study how it is folding by molecular dynamic simulations.
So this high temperature is given by trial and error method, or
no, you can just give a general high temperature like 500 Kelvin at which all proteins will unfold. So yeah, that is just a high temperature that is given. There is no trial and error associated with that.
Okay, ma'am. So trial, is there any case of trial and error in simulation, molecular dynamic simulation?
Trial and error can only be about the time scales if you are not sure. Like this protein is folding in around 20-25 microseconds, but some proteins can take seconds to fold. So in that case, you have to give longer time scale. So that time scale is a little bit trial and error, but otherwise there is no trial and error.
But in general case, we can first apply that flow chart, means what we want to calculate from the flow chart, we can put the time period and then we can do. But if my protein is then also not getting folded, then I have to give the trial and error.
Give longer and longer time scales, yeah. It is always better to start with a longer time scale, because if it is folding in a shorter time scale, then it is then you will observe it in the longer time scale also. So it is generally better to take a longer time scale.
Okay, ma'am.
Yeah, so we studied these two energy terms, the stretching and bending terms. There are some other terms. One is the rotation along the bond. So you know, when you have four atoms, there is a torsional angle associated with them, and rotation along this bond, that can be defined by this torsional angle phi. So there is an angle. So while this bond is rotating, there will be an energy associated with it, because some conformations will be favorable, some will be non-favorable. So that energy is called the energy of rotation along bond. That is given by, again, constant into 1 minus cos n phi. Phi is this torsional angle. And if you add the total energy of bond stretching, bond bending, and bond rotation, you will get the energy associated with bonded atoms. So this is the term for the energy associated with bonded atoms. Now there can be non-bonded interactions also. Like there can be van der Waals interactions. You know the graph associated with van der Waals interactions. Suppose two hydrogen atoms, as they come close together, there is one minimum energy, which is the bond energy. But if you bring them even closer, the repulsion energy comes into effect, and then the energy again increases. So this particular point, the minima of the energy, is the bond energy. And the energy associated with the van der Waals interactions is given by this formula, where A and B are constants and R is the distance between the two atoms. So you can see, so van der Waals interaction is one of the non-bonded interactions. There are other non-bonded interactions also, which I will come to. One is the Coulombic interaction. So if you have two ions, like if you have two ions, one is positively charged, one is negatively charged. Then as you are coming from 5 angstrom to 1 angstrom, they are oppositely charged, so they will attract. And so the energy will decrease as you come close to each other. But if you have two ions with the same charge, like suppose H plus and H plus, then they are the same charged ion. So as you bring them closer from 5 to 1 angstrom, there will be repulsion. So the energy will increase. So this is the non-bonded Coulombic interactions between two ionic species. And that is given by this formula. Q1, Q2 are the charges on the two ions. Rij is the distance between the two ions. There can also be hydrogen bonds where, so ions means you have a full positive charge, full negative charge. In hydrogen atoms, you just have a delta plus on hydrogen, and in oxygen, so this is showing the hydrogen bonding in water. Oxygen has a delta minus, hydrogen has a delta plus, and there is an attraction between them. So it is like the Coulombic interactions, but weaker, because the Coulombic interaction had full charges and hydrogen bonding just has a fraction of a charge. So you can see the dependence of R is also much less. R here is 10 to the power 10 and 10 to the power 12. So very quickly the hydrogen bond energy will fall compared to the Coulombic interaction. And that makes sense because the hydrogen bond is weaker than the Coulomb bonds because it is between a partial positive and partial negative charge. So in the total energy term, you have the bonded interactions, which are the stretching, bending, and bond rotation. And you have non-bonded interactions, which are van der Waals interaction, Coulombic electrostatic interactions, and hydrogen bonding interactions. So there are total 6 terms which determine the total energy of any conformation.
So here we are not using the psi angles, means the angles present between the carbon and nitrogen.
Because in the non-bonded interactions, the angle does not matter. It is only the distance.
No, I mean, in case of bonded also, we were not using that psi angles. We were only using the psi angles.
In the stretching, again, angle does not matter. Angle is only mattering in your like this angular term and the dihedral term.
Yes, means in the bending, the angle matters, but we should also take the means psi angles. Here we are only taking the phi angles. I'm just saying that means.
Oh no, this phi can phi does not have anything to do with that phi psi which we studied. This is a general term for a dihedral angle. So this should actually be represented by something like alpha, where alpha can be phi, alpha can be psi. It can be both. Like this this just represents a general dihedral angle.
So these things are all calculated by programs only, but we can't calculate the same thing.
Yeah, because so many atoms are there, right? So yeah.
And these phi and psi angles, how we will measure this phi and psi angles?
Exactly. So,
but ma'am, the first simulation or first that protein unfolding calculation might be done with the help, might be done manually only, not with the help of softwares.
Yeah, like when the software was built, initially for the software to be built, the program has to be written, right? So generally when someone writes a program, they have to use some model system. So initially they had used some model system to put all these equations into the software. But now you don't have to do any of that manually.
But it's really tough to do manually because how this many angles and this so much angles, and
it is
then
not possible only. It's like not possible. So yeah. Okay. So now we will look at the molecular dynamics process. So first thing is we develop a model system from our understanding of the real system. So whatever structure we have gotten from cryo-electron microscopy or NMR or X-ray, we use that to first build a model structure. Then we apply computer simulation and we compare the experimental and simulation results. If the simulation result is very much different from experimental results, then we improve the simulation conditions or we improve the initial model. And in some cases where experiments are not possible, we perform simulation. So in doing all this, there are some steps which we have to follow. The first step, as I said, is to get a proper structure, 3D structure, which can be done by protein crystallography, NMR, or cryo-electron microscopy. And once we get the 3D structure from this, we can model it in a software. We can create a model of that structure. The next step is to repair the structure. So sometimes, suppose you have found the structure by X-ray crystallography and there is some residue missing here. So you have the electron density map, but there is some residue missing here. So then you have to correct the structure to include a residue which will correctly match with the electron density map. So, or you can have, there can be missing residues. So suppose in when you have modeled, you can see that there is a cysteine 16 and valine 18, but residue 17 is missing. So then you can see that what amino acid will fit in this space, and you can refine the structure to include residue 17. Here it was seen that methionine is fitting, so they have included methionine. So like this, the structure that you initially obtain is repaired. Other than that, if there are steric clashes, that is corrected. If the rotamer conformation of the side chain is incorrect, that is corrected, and so on. The third step is to clean the structure. So the hydrogen is deleted because in molecular dynamic simulation, hydrogen does not matter. Like we will add the hydrogens later, but for now, we will remove the hydrogens to refine the structure. The solvent is also deleted. That again will be added later. And if there are other unwanted factors like extra ligands or extra atoms, that can be deleted to get a clean PDB structure. So this is all about how the initial structure is prepared. The next step is to simulate the behavior of the system over time using specific conditions. So we know from Newton's laws of motions that force is, yes.
In the previous slide, how we will prepare that electron density map?
So this you will get from X-ray crystallography, right?
The one which is already uploaded in the PDF file.
Sorry, I did not understand. What are you saying?
The 3D structures of NMR means not of NMR.
No. See, what is already uploaded in PDB, they will not have all these mistakes. These are the steps for when you are when you have made a new protein and you are you want to you don't have the three-dimensional structure of it. If the three-dimensional structure is already there in the PDB, you don't have to do any of these steps, because you can only deposit a protein in the PDB if all this is taken care of. Okay, so in this case, means which type of protein means like after doing that cryo-electron and all, after that suppose you have a new protein. For that you have done some experiments, and after the experiments you are going to do the simulations. You have not yet deposited any structure in the protein data bank. You have an unknown protein for which you want to do the simulations. So in that case, from X-ray crystallography, you will get the electron density map, but sometimes there can be mistakes like missing residues or the proper side chain has not been included. So that needs to be corrected before any simulation can be done. But if you're taking the structure from the PDB, these corrections have already been done, so you don't have to do it again.
Okay.
Then the the behavior of the system is simulated using different conditions. So we use the Newton's laws of motion. Every atom will have a force acting on it given by mass and into acceleration. And acceleration can be written as the double differentiation of the position with respect to time. This force can also be represented as the gradient of potential energy, negative gradient of potential energy. So minus del V, which is the negative gradient of potential energy, is force. And we can also write force as m d square r dt square, where r is the position. So we are combining equation 2 and 3 to write equation 4, where we are the negative gradient of potential energy is basically mass of the particle into acceleration of the particle, which is given by double differentiation of the position of the particle. So to obtain the movement trajectory of atoms, you have algorithms built in the software which will integrate all these equations. So basically, you know, this left-hand side term, the potential energy, is already being calculated by this entire energy equation. So the software can calculate the left-hand side term, and also it can integrate the right-hand side term to get position and mass. So for each atom, there will be a mass. You have to specify that also, the like it has been written in the program that the exact mass of carbon, exact mass of hydrogen. So using the basic laws of motion and integrating them, the software can find out the position, acceleration at time t for each of the particles. And one more thing is that when we want to select a time step, so time step means that in what interval we want to see the snapshots of the structures. The general rule is to take the time step one magnitude lesser than the fastest motion. So the fastest motion in any protein is generally vibration of the carbon-hydrogen bond, which is 10 femtosecond. Then we should take a time step of 1 femtosecond. So time step means this interval will be 1 femtosecond if we want to look at the vibration of the each carbon hydrogen bond. So depending on, just one second. Depending on what is the motion we want to look at, the general rule is to take the time step between the snapshots as 1/10 times of that motion's time scale. So if we want to look at carbon hydrogen vibration, which has a time scale of 10 femtosecond, we should take a time step of 1 femtosecond. So again, nothing we have to do, the software itself will do, but it is just describing the laws on which the software is calculating. So the software will find out the position and velocity or acceleration of each particle based on the Newton's laws of motions, and it will check, so it will make a list of this position and acceleration in different time steps, and this is how the time step is chosen, 1/10 of the movement we want to look at.
Ma'am, in the case of bond and we want to calculate bond length or bond energy, means we will choose the same time step only.
Yes, correct.
This time step will be 1 femtosecond and the hydrogen vibration will be 10 femtosecond, or this will change that vibration?
No, bond vibration, For carbon hydrogen, it is 10 femtosecond. For larger atoms, suppose carbon sulfur, it might be something else. Then you change the time step accordingly. So yeah.
That is so time step will be 1 femtoseconds only for every.
Oh yes, yeah. Time step will be 1 femtosecond. Yeah.
But this vibration one will change.
This vibration, it can, depending on which atoms you are looking at, that can change.
So this we have to know from the articles only, that which vibration.
Yeah, correct.
And ma'am, this we have to select in the simulation while we are doing the simulation, and means more things we have to select in that simulation process also, leaving this.
Simulation, we just have to specify the time step. These we don't have to specify. It already has the input in the like basic knowledge of the software. All this has been input, like the mass of the common atoms, the time scale of the common bonds. All this has already been input in the software.
And ma'am, that force field also we have to select.
Force field we will select. Yes, that we have to select. I'll come to that solvation and all.
Okay, ma'am. Means after the PDB structure, means my the 3D structure is cleared, means everything is okay for the structure. Then we will go for this step 2.
Yes. This step 2, we don't have to do anything. This like the system itself will calculate.
Yes, but time step we have to select for that.
Yes, only the time step. Yeah.
Yes. Okay.
Then some other files we have to. So we have the PDB file which we have cleaned up. Now we have to include 2 other files, which is the topology file and the parameter file. The topology file basically tells us which bonds are connected. So it will give us an idea about the connection of the different bonds. If 2 bonds are connected, there will be a distance associated with them. If 3 bonds are connected, there are 2 distances, 1 angle. If 4 bonds are connected, there is an additional dihedral angle, so on. So topology file will basically give a complete list of 2 atom bonds. So if you have all the 2 atom bonds, then angle is made up of 2 bonds. Dihedral angle is made up of 3 bonds, and so on. So topology file will give you all those angles. And the parameter file will give us, so we saw the how to calculate the energies. The parameter file will include those things, like again, we don't have to create this. We are just creating the PDB file. Topology file, parameter file, everything the software will create. You just have to give some instructions to the software. There are certain commands which will create these files. It will extract this information from the PDB file itself. So parameter file basically will include the energy terms which we discussed, which will basically define the energy of the system. So these are the 2 files which can be created from the PDB file, and these will come into use when the simulation is being done. Now there can be some non-standard biomolecules, like lysine is a standard amino acid, but ornithine is basically, so lysine side chain, there are 4 CH2 groups and then 1 NH2 group. Ornithine is similar to lysine, only this 4th CH2 group is not there. So it is a non-standard amino acid, or you can say a modified amino acid. So for ornithine, when things like this non-standard amino acid is there, then we this parameter file, the topology file and parameter file, the software has these defined for the standard 20 amino acids. If your system has some non-standard amino acids, then some special commands need to be given. That again, we don't need to go into the details of that. You can just remember this, that inbuilt in the software, it has the ability to account for the 20 naturally occurring amino acids. If you're having any unnatural amino acid like ornithine or AIB, then some separate commands has to be given to deal with the non-natural amino acids. Now, after that, the solvation model has to be specified. Now, there are two kinds of solvation models. One is the explicit solvation model, which uses hundreds and thousands of discrete solvent molecules. So all around the protein, so suppose the solvent is water, all around the protein, you are defining thousands of water molecules. So this becomes a very complicated system, because in thousands of water molecules, you will have hundreds of thousands of atoms, and that many extra position coordinates you will have. So the system becomes very large and the calculations become very slow. This is the explicit solvent model. And implicit solvent model is where, instead of treating the solvent like individual molecules, you treat the solvent as a continuous medium, having the average properties of the overall solvent, and it is surrounding the protein as a whole. So you imagine the protein as a single molecule, and there is a van der Waals radius around the protein, and around that van der Waals radius, there is a continuous medium of the solvent. So instead of specifying thousands of extra coordinates, just a continuous dipolar moment of the solvent is given. This reduces the computational time because all these extra coordinates don't have to be considered. So we have to specify which solvent model we want.
Ma'am, so what we should perform, implicit or implicit model? Which one will be better?
Sorry. See, implicit models are always better because less computation time is required. Explicit models, for one computation, you will need like 1 week or something. So it is not very practical to use unless like you want very detailed
Accurate.
structure like so generally 99% of the cases implicit models only are used. Unless you want a really detailed structure, only then explicit model is used, because the computational time is too high. So yeah, it is generally this that is used.
Implicit models requires, I think, 1 day or minimum maybe
yes,
10
yes.
hours like this. So in the softwares, there is method, means there are steps to choose whether we want explicit models or implicit models.
Correct. And even explicit also, there are different options. Implicit also, there are different options. We don't need to go into the details of that, but that you can choose in the software which particular model you want.
And so to perform this implicit model, after selecting that, various steps we have to do to perform implicit models, or just we have to choose that?
You just have to select, that's it. You have to select and give one command. So the way one of the most common molecular dynamics software is GROMACS. GROMACS is run in Linux. So Linux, you have to give commands. You can't, like Windows, you can just click and do. So Linux does not have that option. So to specify implicit solvent, that is a specific command. You give the command and it will be selected.
What is the command?
So there are like, we don't have that in detail. I'll just show you one thing. Yeah, so I have this slide where, so there are different commands like this, like I'll come to this. So there is this command called solvate. And when you are giving the solvate commands, there will be a pop-down menu where it will give you different solvent models. That also I have in one of the later slides. Okay, so you have to, after solvate, you have to write the solvation model. So then it will add that particular solvation model.
Okay. Right.
Another thing is to define periodic boundary conditions. So basically, like in a crystal, we used to choose unit cell. Periodic boundary condition is to specify a certain boundary in which we are doing the simulations. So it is so suppose you want to calculate the properties of a bulk, crystal, or mixture. You can just select a boundary in which you are calculating the properties, and you can replicate the properties around the boundary to get the and it will be the same in, so it is a homogeneous crystal or mixture. So if you just calculate the properties within one particular square, you will get the properties of the other squares also. So related to the periodic boundary condition, you have this central simulation box. So suppose you find out the dynamics of all the particles within this central box, and you want to find out the dynamics of these boxes surrounding the central box. So what you can do, you can just draw a circle like this, and whatever are the properties of these orange particles which you have already calculated in the central box, these same properties will be there for the boxes surrounding it also. So just by calculating the properties within the boundary conditions, you or the central simulation box, you will find out the properties of the neighboring unit cells also. So this is in case you are looking at a crystal or a mixture. This we don't need to do for a protein, because protein is not a homogeneous molecule as such. So this step is an extra step only if you are looking at a homogeneous mixture or a crystal.
Means what I can't understand this thing. Means what if the protein is means, ma'am, I can't get it.
So in this class we are discussing proteins, so this slide does not have any use in this class. But suppose because even protein crystals are not homogeneous like this. So and protein crystals will not have any dynamics anyway. So this is talking about, suppose you have a liquid so suppose you have some molecules in a liquid which is homogeneous, and you want to study the simulation of that molecule. Then you have to define so you have to define if you want to do the simulations of the entire system, it will take too long. So a unit cell kind of region is defined, which is basically the smallest repeating unit, and you can do the calculations in that particular region, and that will give you the idea of the entire system. So this is something.
In case of proteins, we do not perform.
Yeah. This is not for proteins. This is just suppose you want to simulate properties of a solution of some molecules.
But you said that in this case, protein dynamics will also not be there. So, but protein perform dynamics, means protein are not stationary. So
I said that wrong. What I meant is that this particular periodic boundary condition or central simulation box does not have to be defined for protein. This is generally used when you have a liquid mixture of some small molecules. Just imagine. This is not used for proteins.
And so once I have seen that in the while we are doing the simulation, we perform means we give a box outside the protein. That is this simulation box.
No, that is different. I'm coming to that. Okay. So that is different. That is just wait. So that is different. Like suppose you have the structure of the protein. Around that you add a box. That is to specify the basically we add the box so that we can fill the box with a solvent. So we want to specify a certain volume of solvent which is surrounding the protein. For that we specify the box. That is a different thing. And this, what it is just to specify the smallest repeating unit. In protein you won't have any repeating unit, right? It is not a homogeneous thing. Protein this is generally used only for some bulk liquid, crystal or mixture.
And so liquids are also crystallized.
Yeah, there are something called liquid crystals which has this kind of homogeneity. For that it can be used.
Okay.
It is outside the scope of this. This is just an extra thing. You can remember that an extra parameter has to be included for that.
Okay.
Yeah, now there are certain terms called the phase space. So phase space is basically, it is the collection of all the possible parameters of the system. So phase space consists of all possible values of position and momentum. So for each atom, you will have 3 position coordinates, x, y, z, and 3 momentum coordinates, momentum along x, y, z. So each atom will have 6 parameters in the phase space. And ensemble, this again, this is more related with the statistical mechanics. There are ensemble is basically a collection of states which have different micro state but the same macro state. So this is related to statistical mechanics, and there are different kinds of ensembles. Like microcanonical ensemble is when you have constant number of particles, volume, and energy. So to maintain constant number of particles, there is a lid on the container, and that will maintain constant volume also because the container is sealed. And to maintain constant energy, there is an insulation. So this defines a microcanonical ensemble. So why these are important is basically sometimes you want to do simulations in constant pressure conditions or constant temperature conditions. So then the system, these are just different terms. So then you are doing simulation in a, suppose you are doing it under constant, this is constant pressure and constant temperature. So it is called constant NPT. So the system, there is a constant pressure on the system, and there is constant temperature because it is equilibrating with outside. So these are just different terms which define the conditions under which you are carrying out the simulations. So yeah, so phase space is also, it is referring to the different it is a term which is referring to the list of all possible position and momentum coordinates of all the atoms in the system. So just taking a break from the steps to define some terms. Now it is about, so we saw how to calculate the total energy. Now it is about the potential energy surface. So the basic idea, which was Anfinson's theory, was that the lowest energy state is the protein's native state. So whenever simulation is happening, suppose we are starting from here, the computer will go through all the possible states to find the global energy minima. Local energy minima is also a minima, but it is not the lowest point. So it is called local minima. And global is the lowest point possible. That is the, that's why it's called global minimum, and it is the native state of the protein. So what this potential energy surface, so the computer will basically scan through the entire potential energy surface till it reaches the global minima. So it has all the equations and terms to calculate the total energy. It will calculate that total energy for multiple states, and it will scan through all the states to find the global minima, which will then be called as the native state of the protein. So that is why the energy is calculated, so that it can find the pathway by which the initial conformation is, that is the unfolded state, is coming back to the native state that is folded. So there are certain equations for that. To find the minima, the equation is basically double derivative of the function in terms of x as well as y must both be greater than 0. Only then you will have a minima. And to find a maxima, the double derivative of the function with respect to x and y both have to be less than 0. This is the maxima and minima in the potential energy surface. So you can see that as the computer is scanning through the potential energy surface, this state shown in blue is a local minima. And the high states, these are called transition states. And as it scans, it will go over the entire landscape and it will at last find that this is the lowest possible energy state, which is the global minimum. So that is how basically what the computer will do. It has the energy values. It will double differentiate the energy with respect to the position coordinates. It has the position coordinates also. And it will find out where the minima and maxima are located. And using that, it will find out which is the minima at which the energy is minimum. And saddle point is basically, it looks like this. So when you look with respect to this direction, it is the lowest point. But when you look with respect to this direction, it is the highest point. So with respect to x, it is greater than 0, means it's a minima. And with respect to y, it is less than 0, that is a maxima. So this is called a saddle point. And basically, the transition states are the saddle points. These, when you have a transition state, those points basically form this saddle point. So mathematically, this is what the program does. After it calculates the energy, all the energies of all the states, then it will find out which are the minima and maxima by doing this double derivative with respect to the position coordinates. And once it finds the minima and maxima, it will focus only on the minima, because we want to find the minima energy. It will find which is the minimum at which energy is minimum, and that is the native state. And what is the path that has been followed to reach the native state from the beginning? That will be your folding pathway. So that will help you find the process by which the protein is folding. So this is energy minimization that is happening. Basically, suppose you are starting from this point. The basic rule is that if the slope is positive, let us look at point 1. The slope is like this. Slope is positive. That means the coordinate is very large. So we have to move to this direction. Now, suppose we are at point, sub point here. Then the slope is negative. Slope negative means coordinate is too small. So we have to move in this direction. By doing this, instead, we will ultimately reach a point where the slope is 0. And that will be either a global minima or a local minima. Or so slope 0 basically indicates either a local minima or a global minima or a maxima. So the slope will help find these transition points, and after that we can do the double derivative to find out which is the local and global minimum. So this point, this kind of method of energy minimization. So this is basic. One method is that you go from beginning to end, you go through the entire landscape, but that takes too much time. So the other method is you start from random points. Like I start from 1, I see the slope is positive. That means I have to move in this direction. Then I start from here, I see the slope is negative. That means I have to move in this direction. So instead of studying the entire landscape, I have narrowed it down that it is between this and this region. Like that, starting from random points, you can narrow down the region at which the minima is located. So that is called numerical minimization method. And it is a faster method to find the slope. There are other methods. Yes.
Why we are doing this energy minimization? The graph will be shown in the computer itself then.
Yeah, we are not doing any of this. Like the computer is only doing. Again, I am just, so suppose when the computer is only drawing the graph, but if the computer draws the whole graph from beginning to end, the computational time taken is very large. But if the computer draws a graph like this, which is just starting from random points and converging to a certain point, then the time that the computer will take to draw that region of the graph, which is the important region, will be much smaller. So these are different algorithms which are happening in the computer itself. But this is a faster process compared to the scanning the entire energy landscape.
So in this case, the computer will provide the large graph or the small one?
It will provide only the region of interest. Like if I go back, the computer will only provide, say, fractions. So this entire graph, computer will provide this region. So the different minima it will provide separately.
Means it will show this in the graph format.
Yeah, it will provide the broken graph like this. It won't provide the full graph because you don't need the full graph to find the global minima.
Do you have any photo regarding this, how it will look like?
Like this one only is a photo that was generated. You can see an incomplete graph here, right? If this was extended, you will see some more incomplete graph. So I'll see if I have one where the entire landscape is shown in this kind of, I'll see. I don't have right now. I'll search for it.
And means it will show this graph only. Means do we have to put any commands for this, or it will show itself?
See, during energy minimization command, you can choose which technique you will use. One is the full landscape scanning. One is this numerical minimization technique. Whichever technique you are choosing, according to that, you will get the graph. But the graph is really not important. The minimum point is important. And from the minimum point, what is the structure we will get, that is important.
Okay.
There are some other techniques. We don't need to go into the mathematics of it. You can just remember that there is a steepest, like these are just names of different techniques. You can just remember the latest technique is the Newton-Raphson technique, where it will there is a mathematic so it somehow combines the single derivative, double derivative of energy to very quickly reach from the initial stage to the energy minimum stage. You don't need to go into the, basically as time went on, better and better computer algorithms were developed so that the time it takes to locate the global minima is minimized. And in all those minimization techniques, the Newton-Raphson minimization was the latest technique, where you need very high processing computers to do the technique, but the technique happens very fast because here it is comparing three techniques: steepest descent, conjugate gradient, and Newton-Raphson. Steepest descent is this green line. Imagine R0 is the initial state and this is the final native state. Steepest descent will go in this kind of a pathway till it reaches the final state. Conjugate gradient is slightly faster. It follows only this red pathway. And Newton-Raphson is the fastest from the initial to the final. Here it is taking only one step. And in some more larger proteins, it can maximum take 2 or 3 steps. So as the time went on, better and better minimization techniques were developed so that the computational time taken is minimized. So you don't need to understand more about the mathematics of this. So these are the things that are done before the simulations. You have the PDB structure. If you remember, the hydrogens had been removed. Now we will add the hydrogens back. And we will add a box so that we can specify the volume of the solvent. Then the solvent is added. Here it is water. And ions are added. Like suppose we want it in water which has a certain concentration of some buffer like sodium phosphate. So then we will add sodium and phosphate ions. ions like this often stabilize the three-dimensional structure of the protein. So this is done before the simulation is reached. So each of these steps have an associated command. So again, we don't need to go into the commands because we are just looking at the, we are getting an idea of the technique. We are not going to do any molecular dynamic simulations. So for each of these steps, adding hydrogen, adding the box, adding water, then solvent, there are different commands that are used and different files that are generated. So from the PDB file, ultimately we will reach the .gro file, which is the one which we will use in GROMACS. GROMACS is the molecular dynamics software. So the PDB file is converted into the .gro file, which is put into the molecular dynamics software.
Ma'am, in the previous slide.
This one?
No, ma'am. Yes, ma'am. After why we are is no, ma'am, the previous one only. Ma'am, we have removed hydrogen in the starting. Do we?
Yeah, so we had removed, like while we were making it, just 1 second. If you 1 second. We had deleted hydrogen, right?
Oh, ma'am, so, in the case where we don't have any PDB structure of the protein. Okay, in that case.
If we have PDB, we already have hydrogen. We don't have to add it.
But in the PDB structure, we have to remove, means we have to add solvent and
yes, correct. Some PDB structures, solvent is already there, but that solvent we have to remove because we want to add solvent in that box. So we have to delete the solvent that is there in the PDB, and we have to add new solvent which is restricted to that box we are adding.
So how we will know in that PDB structure the solvent is there or not?
That appears, right? When you download any PDB structure and open it, you can see that there are solvent molecules. I'll show you in the next class. Like, have you used Discovery Studio?
No, ma'am.
I'll show you. Discovery Studio is a very nice software where it shows everything. Whatever is deposited in the PDB file, solvent, ions, everything it shows. So all that has... Just say what you're saying.
I will try next means in this week, and then next week when you will show me, then it will be better for me to understand.
Okay. So from the .pdb file, 1 second. From the .pdb, the .gro is generated after doing all this, which is the GROMACS input file for the GROMACS software, that is the molecular dynamics software. The next, what is done is that different commands are given for the molecular dynamics simulations. So MD run is the final command to do the molecular dynamics simulation, and you will get a graph like this. On x-axis, it is the number of steps. So each step, you can define the time. So step is basically the time interval. So each step means 1 femtosecond. So 500, suppose is 500 femtoseconds. So potential energy is slowly decreasing and it will reach a equilibrium somewhere. That means by that time, you have reached the global minimum structure. You can also carry out these simulations in different conditions, like constant NVT, means constant number of particles, volume, temperature. I was discussing the statistical ensembles. So that was why the statistical ensembles was being discussed, because each ensemble has a different condition, like constant NVT or constant NPT. Then there are certain other commands you have to give. So for these two, again, you will get a potential difference, potential energy curve. Along with that, you will get, so when you are doing constant temperature, you will see temperature is stabilizing out around, if you take the average of this, it will come somewhere around 300. So it might take some time for the temperature to stabilize. but after that, it will be just one straight line. So similarly for constant pressure, then the pressure is also stabilizing after some time. So it is cutting off at 100 picoseconds, means that after 100 picoseconds, the temperature has reached a constant value. Whatever constant value you can specify, like here 300 Kelvin has been specified. So it is taking 100 picosecond for the temperature to come to a equilibrium value of 300 Kelvin. Or here it is again taking 100 picoseconds for the temperature pressure to come to a constant value. I'm not something like 180 or something must have been specified here. So you will get the energy minimization curve, but along with that, you will get an extra curve. If it is constant NVT, you will get a curve like this, and constant NPT, you will get a curve like this. So these are the different outputs after the molecular dynamics simulations.
The NPT and the NVT are same only. What is
NVT is constant volume. N and T are same in both. NVT is constant volume. NPT is constant pressure.
So, ma'am, the file which was generated after that, we will perform this.
It is like sorry, I did not understand your question.
Means, ma'am, that after converting the PDB file to GRO file,
yes.
You said that we have to do. Then after that step, we will perform this minimization and equilibrium.
Correct.
So, this all things we have to do, or just what we need?
Whatever you want to do, you can just do normal energy minimization. You can do this or this, anything, whatever you want. According to that, you have to do.
But how will how will think that what we want?
Like suppose you want to study the effect of temperature on energy minimization. So then you perform, you cannot perform NVT or NPT. You have to perform one of the other calculations, like so different conditions were there, right? So depending on suppose you want to carry out a temperature a simulation in which temperature is changing. So you have to choose one of the conditions where temperature is not constant, like the first one, temperature is not constant, or the last one, temperature is not there. So like that, depending on the system you want to study, it is chosen.
And so that variable temperature, can we choose? How we will choose the variable temperature?
So if you choose this system, which is insulated, so the energy is constant. That means you can specify the a particular, like, wait, how variable temperature, how you can do is, there are two ways of doing variable temperature. One thing is you carry out one NVT at one particular temperature. Suppose 300 Kelvin. Next NVT you carry out at 350 Kelvin. So you can carry out multiple NVTs and put the results together to get the energy minimization at different temperatures.
Where we will put that results, all the NVT results.
So they will come as different graphs, and then you can get the coordinates of the graphs and plot them together in some plotting software like Origin.
Origin. ORIGIN. Okay.
So another command that another thing that can be done is that you can get snapshots of the PDB at different time. So from 0 to 100 nanoseconds, suppose I am saying that I want snapshots of the PDB at 20 nanosecond interval. So at 0, 20, 40, 60, 80, 100, it will generate separate PDB files depicting the structure of the protein at that particular time. So then after you have all these different PDBs, you can see that how, so that is one you can, that you can get, and you can find out how the protein is moving with respect to time. And there are some values which we can calculate. This RMSD, RMSF, I will discuss in the coming slides. Before that, one more thing that can be done is this command called trjconv, which is a post-processing tool. So sometimes what happens is that we want to visualize the trajectory, meaning how the molecule is moving in a software, which is the VMD is the best visualization software to look at the molecule trajectory. That is the path of movement of the molecule. So in VMD, there is a square like this, and the trajectory has to, sometimes it appears like this. So basically what all this thing that you are seeing, initially it can come like this, and then a command has to be given, trjconv, which is a post-processing tool. So it will alter the trajectory so it properly fits the box has moved here. The box will be around the protein properly. So basically what this is, you can imagine this as a superimposition of this is basically showing how the protein has moved in all this time. So sometimes the result can be like this, where in the observation box, we are getting 1/4 of the protein from 4 different protein molecules. But we don't want this. We want one whole protein molecule to fit inside the box. So this blue box will come around the protein properly, and that is done by a post-processing command called trjconv. So basically, the protein can diffuse through the unit cell and may appear broken. So this looks like the protein is broken up into 4 parts. That can be corrected using this command. It is basically like a post-processing tool to better our visualization of the protein. And now we will look at the different things that can be calculated. One is the RMSD. So RMSD is basically the deviation of, so RMSD can be calculated for each atom, the deviation of the atom from its, so delta I is the distance between 2 different positions of the atom I. And if you have, say, 100 structures, you will have, if you have n structures, you will have n values of delta I. And if you divide that by n and take the root, that is the root mean square deviation of that particular atom. So that can be plotted with time. So suppose you have 2 proteins, one in which the RMSD, you can see RMSD, initially it is low, but then it is becoming high. So somehow the protein is, initially maybe it was folded, and as the time is going, it is becoming, initially maybe there was a lot of secondary structures where, as you know, secondary structures do not move very much because they are stable. So the RMSD was low initially, but after around 20 seconds, probably some of the secondary structures are unfolding, which is why the RMSD is suddenly becoming high. Because in loops and unfolded regions, you know, there is a lot of movement. So the RMSD will be high. Whereas the second protein shown in red here, RMSD is always high. So that means this protein must have a lot of flexible regions. So the RMSD can be calculated and plotted to find out an idea about the dynamics of the protein with time. Another is the RMSF. So these are all, you don't have to manually calculate it. This command rmsd.xvg, it will generate this kind of a graph. Then there is something called root mean square fluctuation, or RMSF, which calculates the fluctuation of C alpha atom with average structure. So at time t, t1, if the C alpha atom is at x1, that is subtracted from the average position of C alpha, and again it is squared and divided by the number of structures or time that you have. with root. So root RMSD and RMSF are very similar, except that RMSF is only for the C alpha atoms, and RMSD can be for any atom.
Ma'am,
And yes,
in the case of RMSD, the unfolded protein means we are calculating the folded protein to the unfolded, right? Or we are calculating the means we are selecting the unfolded protein to folded protein like this, or means you have said na, that while calculating RMSD we are finding the deviation of atom from different positions. So is it about the folded protein or is it about the unfolded protein?
It is at any, see, whatever time, so this is the time interval on the x-axis. So at that time, whatever is the structure of the protein, it will calculate the RMSD with the average structure. So it is not between like RMSD can be calculated for both folded and unfolded. So it is just like that is what the difference of the atomic coordinates from the average atomic coordinates is what is being calculated. So yeah, like I am not understanding your question actually.
Ma'am, means my concept is not getting clear because we have to calculate the RMSD, right? So we are calculating the RMSD of the atoms, or we are calculating of the whole protein?
Like it is the atoms, but it is added over all the like okay, that is actually a good question.
Explain with an example.
You are right. I'll have to find this out because according to the RMSD definition, it is atom wise, but here it is not specified. So I have to actually find out about for what the RMSD has been calculated here.
And
I'll find out and let you know.
Miss, and the points which is there in the graph, is it representing the protein or is it representing the atoms?
Because see, what I think is it is representing a particular atom, like RMSF is representing C alpha. So it is representing a particular atom. RMSD probably is also representing a particular atom, but you can specify which atom you want the RMSD for, because you that is what you are asking, right?
Yes, ma'am. Means I am not getting clear about what is this RMSD about proteins. Means you said not that while the unfolding of the protein gets many structure. So is it about the atoms present in one particular protein structure, or is it the means atoms means one by one atom?
What I understood is that it is RMSD is for a particular atom. Like say I choose the amide nitrogen. So from 0 to 100 nanosecond, the amide hydrogen is occupying many different in each structure that you have from 0 to 100 nanosecond. The amide hydrogen is occupying different positions, and you can calculate the RMSD of that particular atom between the different structures.
So, ma'am, after yes, the means in case of the protein, there are various atoms. So the atom is changing its position, and then the RMSD is being calculated, or the means the average of all the atoms. Suppose we are calculating of the amide bond, as you said. So it is about all the atoms, and then they are taking the average. Means average is of two types, right? So means
no, I understand what you are saying. I have to find out actually. I am not sure about this.
And means
I will find out and let you know.
And we are doing everything, means we are till now what we have done is we are trying to get that folded structure to the unfolded structure. After doing that, we are calculating this RMSD. So RMSD is about all the unfolded structures, or means it's very much complicated, right? We can't
See,
say
what I understood is that we are doing this minim.mdp is giving you the energy minimized structure, and then the energy minimized structure itself is moving. So these are the different snapshots you have taken of the energy minimized structure. And so that means of the native structure itself. So first, from unfolded, you are going to folded. So the energy is getting minimized and it is reaching like it is becoming constant when you have reached the minimum energy structure, which is your native structure. Once you have reached the native structure, then you are getting different snapshots of the structure as it is moving, because in the native structure also there is some movement. So as it is moving, you are taking different pictures of that. Yes, you can see what is written here. It is written C alpha RMSD. So RMSD is calculated for one particular, I'll just repeat what I said. Energy minimization is done, and from the unfolded state, you are coming to the lowest energy state, that is the folded state. So in this process, while coming from unfolded to folded, taking different snapshots. Now within these snapshots, RMSD of the native state, that is the final structure, is here C alpha RMSD is written. So C alpha in 0 nanosecond is compared with C alpha of the final nanosecond. That is your RMSD of the at 0 nanosecond, what is the RMSD? Then at 20 nanosecond, RMSD will be something else. 40 nanosecond, it will be something else. So that is what I am understanding, is that it is comparing at each time the position of one particular atom, it is comparing with the final structure to find the root mean square deviation.
So ma'am, is it about all the C alpha atoms or specified C alpha atoms? Means is it about all the
it will be all the C alpha atoms, not one particular. You can just choose, if you want C alpha, it will calculate for the C alpha of all the amino acids. If you want amide nitrogen, it will calculate for the amide nitrogen of all the amino acids.
So do we have to give some comments related to this?
You have to so these commands are common, but then RMSD for which atom you want, that there are different keywords. They are not mentioned here. That will be different for different atoms.
So with respect to 100 nanoseconds, the 0 nanoseconds is calculated, and rest of the is done the same way. So in 100 nanoseconds, it will be calculated as the 100 nanoseconds only. And but it will not be like not linear, a constant graph. Miss, we think that like 100 nanoseconds is calculating with 100 nanoseconds, so
it will be 0 at 100 nanoseconds.
Yes, yes, but this will not be done because I think we are calculating of the overall all C alpha atoms.
Correct.
Okay.
I will still confirm. This is what I have understood, but I will still confirm and let you know.
Okay.
Okay. So, and
RMS. The slides much more means how much slides? How much slides?
We won't be able to cover all the slides. I will just I guess rest. What I can do is you go through the slides and you tell me which slides to explain. Maybe I can hold a separate session to explain those slides. There are many more other slides. We can just go to the questions for now, and you can tell me which slides you are having problems understanding, and I'll explain those. I'll just come to the questions for now. So molecular dynamics simulations are based on which one?
Newton's equation.
Yeah, that I had discussed, that based on these equations only, the software is programmed to calculate the energies, positions, and momentums of each particle. Then it is when we convert the .pdb to .gro, which file will not be generated. This you will not remember because it is not given properly, not discussed in detail. So I will just discuss. From .pdb, when you go to .gro, .gro is being generated at the last step. Topol.top, you have to input that. This is basically a topology file. We studied that topology file is where from the PDB file, we have to generate parameter file and topology file. Topology file is about the connection between different atoms. So that has to be generated from the PDB because that will you need the topology and the parameter file to specify the GROMACS file, because topology file will tell you about the connections between different atoms, which will help you define how that bond length and everything is changing. So topology file is also being generated. This is a file position. So POSRE.ITP, it contains information about positional restraints. Like suppose you have a cyclic molecule like proline. Proline, you know that the bonds cannot rotate. They are because in a cyclic molecule, you cannot rotate the bonds. They are restrained in that position. So this particular file, POSRE.ITP, it contains information about positional restraints. That is, which positions are restrained and cannot rotate freely. So these three files are all needed while generating the GROMACS file. And what is this .xtc file? We will just go back. .xtc is being generated after the molecular dynamics simulation. So basically, it was asking from PDB to .gro, what are the files being generated? .xtc is generated after that during the molecular dynamics simulation run. So you don't have to remember all the commands and everything, but you can just remember these file extensions. That .gro is the GROMACS file. .mdp is the energy minimization file, and so on. So that is what this.
But the question is wrong, because .gro file, how the gro file will be there?
Yeah, it is. Question is wrong. I agree. So it should write PDB to GROMACS input file or something should have been the question.
Yes.
So yeah. So you can just remember that GRO is the GROMACS input file. Topol is the topology file, which contains information about the atomic connections. POSRE.ITP contains information about positional restraints. That is, which atoms are restrained. So all these three are there while you are going from PDB to the GROMACS input file. But .xtc is generated after the simulation. So the answer is D, where this is not generated in the first step. This was not discussed, but I'll discuss it in the question. In the topology file, there is a term called RESNR. What does it stand for? I think you can guess it. From the looking at it, can you guess?
Residue, it's confirmed.
Residue and NR for?
Number.
Yes, correct. So B is residue number. So many of these you can look at it and guess. So there is this term RESNR. It specifies the residue number. So yeah, that is part of the topology file. It will tell you that in residue number 1, which atoms are connected. In residue number 2, which atoms are connected. Like that, it is mentioned in the topology file. Phase space is a how many dimensional space. So if you remember,
3D?
3D? No, actually it is 6-dimensional because you have position, momentum, and so you have ignored, so you have XYZ position, XYZ momentum. So if you include all this, it will be a 6-dimensional space. So it is defined like that, but I also have like, why is it's just XYZ, right? So it should be 3-dimensional, but I think what they mean by dimension here is not that 2D, 3D we know. By dimension, they probably mean the number of parameters, which is 6.
No, ma'am, it's the dimension only, because distance and means I think Q will be the displacement and P will be the momentum, and these two are the vector quantity.
Okay, so they are talking in terms of that dimension, yeah.
Yes,
so
maybe means I am not sure, but I'm just guessing.
Yeah, so it was written that it is 6-dimensional place because there are 3 positions and 3 momentums. So yeah, I guess because it is a vector quantity, it is being defined like that. Yeah, yes, you're correct. Then it is saying which physical property is constant in microcanonical and canonical ensembles. So for that, you just remember that slide where all the ensembles was being shown. So microcanonical was constant NVE, where E is energy, and canonical was constant NVT. So in both, the common is NV. So constant number of atoms and volume is constant in both. That is option A. So again, this is something you have to memorize, that wherever that graph with all the ensembles which was shown with the names, you just have to memorize that. Then it is asking what is the condition for minimum, maximum, and saddle points in potential energy space. So here, if you have to remember this, for minima, the function, double differentiation of function with both x and y is greater than 0. For maximum, the double differentiation with respect to both x and y is less than 0. And for saddle point, 1 will be greater than 0, 1 will be less than 0, which is there in option B. So again, you just have to remember these conditions which define the minimum, maximum, and saddle point in potential energy space. Then it is asking that which of the following is known as bonding energy term. So can you say this?
Is it carbon bond term?
Yeah, see, there is nothing called carbon bond. So this term is only wrong. So actually, there is nothing called carbon bond. So this is wrong. Hydrogen bond is non-bonding. So this is wrong. And there is nothing called distance bending. So this term is also wrong. So the only correct term is this angle bending term, which is this one. If it was written carbon-carbon bond, that will be also correct. But carbon bond, there is nothing known called carbon bond. So this option is confusing, but it is wrong. And so here the only term, so in the bonding energy term, you have 3 terms: angle bending, bond stretching, and rotation along bond. And among these 3, the only one in the option is the angle bending. So it is option C. Again, this I have not discussed. In the implicit solvent models, so I discussed explicit and implicit solvent models. In these, there are different solvent models, which we don't need to go into the details of, but you can memorize them. So implicit has 4 different models. Explicit has some other subclasses. So which is not implicit solvent model? If you look at this, implicit Poisson-Boltzmann is implicit. So this is out. Generalized bond model is implicit. So this is also out. Karplus model is also implicit. So this is also out. So polarizable water model is explicit. So this is the only explicit model. So that is option C. Again, this is something you have to memorize. If you want to look into what each of these models are, you can do that, but that is outside the scope of this class. You can just memorize these subclasses. The topology file can specify what? Can you try this? If you have to choose only one option, which one will you choose?
Options connected.
Sorry? It's the first one actually. So topology file, it will tell you about the bonds which are two atoms connected. From the two atom connected bonds, you can, so if you know the bond, then when two bonds are connected, you will know the angle. When three bonds are connected, you will know the dihedral. So the most important thing specified is the bonds, which is option A. And this is what is the post-processing tool for analysis. This was discussed in one slide. It is trjconv. This command is used for post-processing to visualize the protein as a whole. So again, there were so many concepts discussed, and I couldn't cover the later concepts. So you go through the slides and you can just mail me which slides you are not understanding. So in the next class, I will explain those slides and then I will discuss the next class points. So I think I'll end the class here. And again, you go through the slides, you go through the video, and you ask me your doubts. And I will look into the RMSD once more and confirm that what I have said is correct. So that I'll do from my side, that the RMSD I will confirm once. Okay, I'll end the class here then. Okay, thank you,
ma'am.